So suppose if you have fixed your interval like at 0.5 hour, 1 hour, 2 hour, 4 hour, 8 hour, 12 and 24 hours. So you cannot take a blood from a single animal. The reason is, from a single animal, if you will take a 0.5 ml, the approximate the total blood which you will take in one day will be approximate 4 to 5 ml, which is not allowed by CPC SEA, as well as your protocol will not be approved by institutional animal ethical committees. So what you can do here, you should select a more number of animal. So in if you are having a more number of animal, likely I will tell you, suppose if you want to do pharmacokinetic analysis of your one formulation, you have a single formulation. So what you will do, you can select minimum 8 animals. So suppose if you are selecting minimum 8 animal, your total intervals are approximate 7 to 8. So what you will do, you will take a animal blood 0.5 ml from a single animal once a day. So it will be beneficial because it will not be against ethics and your protocol can easily be completed. So this is very much necessary that you people should know that you should never take a animal blood more than approximate 8% or exceeding limit is 10%. So what will be the dose levels? So many a times you know that during the research paper or whenever you do research, you maximally select low dose, intermediate, high dose. So you should know firstly that whether previously it has been reported that you are selecting a 5 mg, 25 mg, 50 mg like that, or you are selecting a 25, 50 or 100, or you are selecting a 100, 200 or 500 or 250. So whatsoever the you can say schedule you are following, either this 5, 25, 50 or like that, if you know that these dose has been well reported that they do not produce any significant effect, so for the herbal drugs you always go for either medium dose or a higher dose. And if you know that this drug is potent and its lowest concentration can be toxic, then it is better that then you should go for the in vitro analysis firstly. You should calculate IC50 value, and from the IC50 value you can easily drive ED50. So what will be the approximate ED50 of that particular drug? Either you can go for in vivo analysis in the animal. So basically the preference dose are low dose, either maximal if you see during the research it is found to be non-toxic, and we always go for the analysis between intermediate and the highest dose of the drug. So this is a rectifying factor on the study to be considered. The things are protein binding, tissue uptake, receptor properties and metabolic profile. So basically it depends that which type of research you are doing. Either suppose if you have synthesized some of the derivative of diazepam or synthesize some of the derivative of COX inhibitor like that. So the things, this type of things are previously well reported. But suppose if your drug is new and you want to explore a new molecular mechanism, what you can do, you should choose some of the biomarkers. So you choose that your drug is new and you are going to find the new therapeutic potency of the drug or efficacy of the drug, then you should select some of the biomarker XYZ. And from these biomarker analysis you can find that your drug at a molecular level do some changes in the cell and tissue levels. So that is very important. And either you can go for the receptor properties. So basically this receptor properties can easily be analyzed at the initial level whenever you will perform some docking or some other studies. The second thing which is very important, that is route of administration. This is a big lacuna in many of the research or in many of the researchers. You know that they select sometime few drugs. Suppose I will give example to you that suppose if you have planned one of the research that you want to do that research in your lab, then how you can select the route of drug administration. The first parameter as per the, you can say, OECD, and as per the ICH, International Council for Harmonization and Organization of Economic Control and Development. So what the basic question they ask, suppose you are going toxicokinetic analysis, the route which you will select in the animal that will be similar to the human. So don't use any drug if we are giving in human by oral route and you are giving in animal by IP route. This is not a rational thing. So route should be matched. Then it is very easy. Then your protocol, your research paper can easily be published, and there will be no any major commands you will follow there. So the thing is, if you are doing inhalation study, topical, parenteral studies, the basic thing you keep in mind that route of drug administration should be selected very carefully, and it will resembles clinically to the human being. So it is important because many a times for the herbal drug we select a oral route of drug administration, and as you know that herbal drugs or herbal antioxidants or prebiotics, probiotics which are available in the market, we always take by the oral route. So what is the next thing, that is a metabolite. So sometime you know that your drug is converted into metabolite, and it is not easy that you will assess this metabolite in the plasma. So basically this is required if you know that our drug produce some of the toxicity in our body. So likely you know that many of the drug, morphine which is converted into a 6 morphine, or you can say apomorphine, like that. Similarly, you know that in your body, levodopa is converted into dopamine in the brain, and as you know that paracetamol is converted into one of the metabolite which is called as a NABQI, N-acetyl benzoquinone, which is toxic for the liver. So in certain cases, if you know that the metabolite of the drug is toxic, then you should find the metabolite and you should analyze the metabolite by using HPLC, HPTLC, whatsoever, mass spectroscopy, or any other, you can say NMR analysis. So the thing as per OECD and ICH is that if your metabolite concentration is more than 5%, then it is must that you should analyze the metabolite concentration in the blood of that animal, in the plasma of that animal. So it is must that you should do the metabolic analysis. So the next thing is statistical analysis. So basically the statistical method which you are using, it depends upon the data, type of the data. Either your data is normal distributed, abnormal distributed, either you are calculating mean, median, mode, or you are having some groups of animal, you are going for one way analysis, two way analysis, like that. So according to the data which you are taking from the animal study, you should analyze your data. So today in this lecture I have discussed with you what are the basic principles of toxicokinetic analysis, how you can perform toxicity in animals, how you can give a drug to the animals, and in the next lecture, I will discuss with you which type of animals are particularly preferred for some type of the studies. So I will discuss, you should like, subscribe this Pharma Learning channel. You all are welcome. Thank you very much.